Hi, Emilio. Can you hear me? Emilio? Are you
I can
there?
hear you.
Great. Thank you so much for joining us. We just need you to get on screen here.
Yes,
There you
there
are.
is try.
Now we can also see you. So thank you so much for
Okay,
joining.
great.
Are,
Thank you
are you
very much.
are you able to share your slides?
I should be. Okay, can you see my slide?
We can see your slides. Looks
Oh,
great. Thank you so much.
perfect.
Whenever you're ready, you can start.
Thank you very much. Thank you very much for the invitation, and I'm very sorry if I couldn't join in person, and I'm glad that you gave me the opportunity to speak virtually through Zoom. So today I would like to tell you a story um, about cataract. You know that aging comes with a lot of aging-related diseases, and cataract is one of them. You see that cataract it's something that affects the eye lens that becomes opaque, and unfortunately, if this happens, the sight is almost lost. The only treatment is surgery. And this is something that, of course, extremely frequently in elderly people. And you know that the elderly population in the world is getting larger. So clearly, there is an epidemic of cataract in these days. And so cataract is usually thought to occur because of senescence happening in the lens eye. And this senescence causes, of course, the classical responses. We have inflammation going on, cells that don't behave as they should. And in principle, this leads to the opacity of the lens and blocks sight. So we don't know much about how cataract is developed and why this is occurring in the elderly, and why this what the genes are that actually drive senescence in the elderly. And almost by chance, a few years ago, we stumbled into a gene that we were studying, and we stumbled in patients that were knockout for this gene, so they didn't express this gene at all, and these were young people, teenagers, and these teenagers showed some signs of increased or actually accelerated aging. And among the various phenotypes they showed, including stroke, the bone aging, they also showed precocious cataract. So this cataract was taking place in young people. And the gene we were studying was PI3 kinase. And this was very unexpected, if you want, because a loss of a PI3 kinase was causing some kind of accelerated aging. And I'm sure you all know that the PI3 kinase pathway is usually linked to age control and aging, but actually the link is a positive one. We heard before that mTOR is connected to aging, and if we block mTOR activity, we can reduce aging. And usually mTOR is controlled by the PI3 kinase pathway. And in mice, for example, insulin activates PI3 kinases, and this is controlling FOX activity, which is connected to lifespan. And we know that in C. elegans, for example, as we heard from the previous talk, C. elegans is a wonderful model for lifespan related genes, and all of the genes of the PI3 kinase pathway, when knocked out, they actually increase longevity. So we were dealing with a very unexpected mutation, a mutation of a PI3 kinase that was doing the opposite of what was expected. It was not increasing longevity, but actually it was actually accelerating aging. But I have to say that the PI3 we were studying, that is called PI3 C2 alpha, is a special PI3, and it's not exactly the same PI3 enzyme that we know associated with mTOR regulation and FOXO regulation and shortening of the lifespan. So this PI3 makes phosphorylated phosphoinositide like all other PI3 kinases, but doesn't make exactly the same phosphorylated phosphoinositides as the classical PI3 kinases we always heard about that are connected to longevity. And this is actually called a class 2 PI3 kinase. And this, it's a family of genes. Actually, this class 2 PI3 kinase is expressed in C. elegans. We don't know precisely what the mutation of this specific PI3 kinase is doing, but certainly is not a DAF gene. It's also present in Drosophila. And in the humans, well, we have 3 different genes. And the patients I was alluding to before were patients where the PI3 kinase C2 alpha gene was knocked out. And PI3 kinase C2 alpha makes some phosphorylated phosphoinositides like PI3P and PI34P2. PI3P is made in internal membranes inside the cell, and PI34P2 is usually made at the plasma membrane. But the classical PI3 kinases actually make other lipids. For example, PIP3, that is a different thing. And so we wanted to understand a little bit more about the function of this unusual PI3 kinase. And we went first to look into cells and we analyzed its expression pattern. And we realized that it had a very peculiar expression pattern during mitosis. So it became extremely well localized in the last, very last time, the very last phases of mitosis, cytokinesis, and the completion of cytokinesis, abscission. And in cytokinesis, this PI3 kinase C2 alpha appeared nicely localizing with the mid-body, which is an organelle that is indeed involved in the cleavage of the intercellular bridge between the 2 daughter cells. And so apparently PI3 kinase C2 alpha had a role in the final cut between 2 daughter cells. And indeed, if we looked at cytokinesis in cells with the normal amount of PI3 kinase C2 alpha or cells from the knockout patients, well, we could see a strongly delayed cytokinesis process, and you see that this strongly delayed cytokinesis is accompanied by binucleated cells. So the tendency is to refuse after this failure of mitosis. And this is linked to the triggering of a senescence program, because we realized that these fibroblasts lacking PI3 kinase C2 alpha showed increased SA beta gal expression. And they also showed significantly increased P16 expression, indicating that this effect was connected somehow to the establishment of senescence. So to have a better in vivo model, we shifted to zebrafish, and we knocked down PI3 kinase C2 alpha in zebrafish. And as you can appreciate, we have a lot of cells stalled in cytokinesis, particularly in the eye lens, exactly where we were seeing the major phenotype in humans. And so when we went back to the eye of these knockdown zebrafish, we could detect a strong enrichment of senescent cells exactly in the lens. The eye was kind of smaller, we had less dividing cells, but the striking effect was indeed specifically in the eye. So we had some senescence everywhere, but the most striking effect was really in the lens. And in the lens, we also had increased expression of P16. So this was accompanied by the activation of the pro-survival pathways and of the classical SASP program. They express several cytokines and inflammatory agents. And this was not only true in the lens of the zebrafish, but this was also detected in human fibroblasts, and actually even in human lens cells put in culture. So indicating that likely the phenotype seen in patients lacking PI3K alpha was indeed triggered by some function of PI3K alpha in cytokinesis, and we also could detect cataract in this knockdown zebrafish. We were of course wondering whether this effect was somehow related to abnormal embryonic development. So we generated inducible knockout mice that were reaching adulthood normally, and then we treated these mice when they could express tamoxifen inducible CRE with tamoxifen in the eye. And after 4 to 6 months, as you can appreciate, all of these mice started to develop cataract, very similar to what seen in patients. So this was clearly not related to an embryonic phenotype, but was indeed acquired like it is in the elderly. So what was the mechanism? So we realized that PI3K alpha was producing this special lipid PI340P2 at the cytokinetic bridge. And we hypothesized that this was important for the organization of the complex that actually calling escort proteins cleaves the cytokinetic bridge. But the problem is that the literature is showing that there are many pathways that do this job and call escort proteins like CHMP4B to the cytokinetic bridge. And one protein is, for example, ALIX. But we realized that exactly in the lens, this redundancy is absent. And as you can appreciate, in the lens of fish and mice, we detected a significant decrease in the expression of the ALIX protein. So in principle, this is the cleavage of the intercellular bridge is a key important biological process. So it's a process that needs to be resilient, so there is redundancy. But apparently for some reasons that we still don't know, in the lens this redundancy is lost and the ALIX protein is significantly less expressed. And this is why the loss of this PI3 kinase, it blocks the recruitment of VPS36 that indeed, as you can appreciate from this movie, actually goes exactly to the place between the two daughter cells where the cleavage of the membrane needs to be organized. So we developed again knockdown fish for this second protein that we found binding the lipids produced by PI3K alpha, and as you can appreciate in this knockdown of VPS36, we again saw increased numbers of cells stuck in cytokinesis, and once more we detected a strong senescent response with again a very strong localization within the lens rather than the eye itself. And again, we could see P16 being upregulated specifically in the lens, where likely redundancy is absent. It's possible that the lens needs to avoid ALIX because of the transparency needed. We don't know yet, but these are all hypotheses. So the mechanism is that PI3 kinase produces a lipid. The lipid recruits escort proteins, and escort proteins induce the cleavage of the cytokinetic bridge. In the absence of PI3K alpha, we have a delayed cytokinesis, and usually delayed cytokinesis causes refusion, and refusion is also something that triggers aneuploidy. Of course, tetraploidy is the starting point for subsequent aneuploidy when cells re-enter their cycle. And this is likely why we have increased senescence in the absence of this specific PI3 kinase, and because redundancy in the system is lost in the lens, this is clearly triggering a specific response exactly there. And this is nicely taken by the news and views that accompany our paper in Science a few months ago. But the key is that we still don't understand in detail why the senescence response is triggered and what the mechanism is. So lately, we collected some new data, some new unpublished data, indicating that probably the usual suspects are indeed involved in this process. And again, if cells fail cytokinesis, they refuse, and when they re-enter the cycle, they have to start with an exaggerated amount of chromosomes, but also a duplicated amount of centrosomes, and this leads to abnormal spindle development, and the consequence of this is usually the emergence of micronuclei, and the macronuclei are usually triggering the activation of the cGAS STING pathway, which is one of the key driver of the SASP response in senescent cells. And actually, if we block the production of the substrate taken by PI3 kinase C2-alpha to make its famous lipid, well, we see that the cGAS STING pathway is activated, and we see cGAS being localized in the micronuclei. So we are now actively trying to figure out more details of this mechanism because we know that cGAS is potentially druggable, and maybe we can find in the future a new strategy that besides surgery can block cataract in the elderly. And I would like to stop here by mentioning Federico, Quai, Lorenzo Prever, those guys that actually did this work, the funding agencies and the various collaborators around the world. And I would like to thank you for the kind invitation and the patience of listening to me over Zoom. Thank you very much.
Thank you so much, Emilio. That was a really fantastic talk. Do we have any questions in the audience? Maybe I can start. So we know that in some of the premature aging diseases, we also see cataracts, and they are associated with genome instability also. Do you think this is a completely universal route for cataract development, or is it?
I think it might be actually, because we know that, for example, mutation in the YAPTAZ signaling pathway causes cataract, and now we recently saw in Nature papers connecting YAPTAZ effects with cGAS activation. So it might really be a kind of a common mechanism that in the future we might be able to fight with cGAS antagonists or let's say other drugs that reduce the senescent response detected in the eye. On the other hand, it's extremely interesting to find out what genetic traits are predisposing to cataract development. So we're actually trying to understand if patients with higher risk of cataract development actually express a lower PI3 kinase C2-alpha. And in the paper, we put some preliminary data indicating that this is the case.
Really super exciting. Do we have more questions here in the audience? Everybody just ate, so I think they are digesting right now, Emilio. But That was really a fantastic talk. I really enjoyed it very much, and thank you so much for joining.
Thank you very much. And I will follow you on YouTube.
Thank you so much. Bye-bye.
Bye.